All right, so just in just a few seconds, you're going to know how to answer this college exam question. So transcriptional errors, when we are copying from our DNA template to create mRNA molecules, there are several mistakes that the RNA polymerase can make. And there's also ways that our body corrects these mistakes, but for now I'm just going over the mistakes. So first one is this silent error. This is when we have a nucleotide swap. The mRNA puts in the wrong nucleotide, but it doesn't really affect the protein. And that's because of something called codon degeneracy, which is really cool. So basically, all of our amino acids have multiple codons that code for them. And so if we accidentally swap it, most of the times the third nucleotide, we still get the same amino acid in the end. So for example, for phenylalanine, if we were to swap the U for a C, we'd still get phenylalanine. Next is missense mutations. This is very similar, except for it does change the amino acid. And there's conservative and non-conservative. Conservative basically means we change the amino acid, but it basically has the same function as the other amino acid. Oftentimes it's the same type of amino acid. And so there's not really any effect on the protein. Non-conservative is just what it sounds like. It will have basically a big effect on the protein, and that's not good. That's a much more serious error. Nonsense mutations. These are when we change a codon into a stop codon. And so we stop the mRNA sequence early. So for example, we changed UAU for tyrosine to UAG, which is obviously one of our stop codons. Then we have our frame shift errors. This is when we skip or add a base. This is even a more detrimental effect, as you can imagine. If we just happen to add an A where it didn't need to be, every single codon down the line, you can imagine, would be totally affected and different. And finally, we have a read through, and this is where we just fail to stop at the right termination sequence. So hopefully this was helpful, hopefully a good overview, and yeah, see you on the next one.